Going to go ahead and start recording. I'm also just going to mute everybody just in case there's some crazy noises that come up in the background, but you're welcome to unmute yourself if you have a question or anything. All right, so today we're going to finish talking about membranes, and then we'll just transition very smoothly into talking about how we're able to transport substances in and out of the cell through this lovely membrane that we've constructed. So that's kind of where we're heading today. Just a reminder, if you haven't done so already, and I'll be honest, I haven't checked um, all of your team accountability forms for assignment 2, so you are reflecting on how well your team worked and what you may want to change or do differently as you're working on assignment 3. So, please make sure you take some time to fill that out. They will be used, so I can kind of help your teams as well if there's any group struggling. And then pretty much today is a lecture day. Wednesday, we will do an IRAT, TRAT, and Friday we have our little TBL activity looking at cell transport. All right, so today where we are, and actually it doesn't look like I stuck it on this list. We're looking at really the cell membrane, and we will look at how saturated and unsaturated fatty acids kind of are mixed when we're building this membrane to allow for it to function in various temperatures, really is one of the main reasons for that. So we will apply a lot of what we did with the lipids from last week into building our membranes for this week. And just a reminder, we did this slide on Friday, but what we were trying to accomplish was a barrier between the inside and outside of a cell, right? So to create its own little enclosed environment. We discussed that both sides of this membrane were going to be exposed to water. And so that really impacted what type of material we could use to construct this membrane with. But we also need to be able to control what can come in and out of this membrane. So today we're really going to focus a lot on how we're able to control some of this stuff. All right, so we said we really needed a combination of a lot of these different types of macromolecules. Our main component is going to be actually a phospholipid. So it's going to be that lipid that we looked at. I think I have a picture of it on the next slide, where the head is going to be hydrophilic. It's going to want to interact with water, where the tail is going to be that very common carbon hydrogen tail that makes up lipids, right, which is very hydrophobic. We will also implant within this membrane some carbohydrates as well as lots of proteins. So we actually need all of these things in order to accomplish our goal. So let's just review our favorite amphipathic molecule, right, the phospholipid. So we noticed that the head of this is hydrophilic. It has specific charges, as well as we will see some of those polar covalent bonds in several areas. So giving this an overall polar aspect, making it wanting to interact with water. And then it will have these tails, and the tails we'll see can vary in length as well as their saturation levels. So sometimes we will see those double bonds, which will create some kinks in these phospholipid tails, which is actually going to help give us some different support and fluidity in our membrane. All right, so this is just kind of a review from last week. So now we're going to get into something a little bit new, and I know this slide is a little bit busy, but hopefully it'll allow us to kind of walk through some different aspects here. So on the left-hand side, I have a couple pictures, and the top picture is showing us some phospholipids. I know they look like little lollipops, but these are our phospholipids. And what we see is how they react if we were to just dump a bunch of phospholipids into a beaker of water. So the tails themselves are all going to point inward and the heads are all going to be on the outside. So we have to think of this image here as kind of a slice out of a three-dimensional kind of sphere structure. So I always think of basketballs when I'm doing this one because they have the little um, dots, right? The raised dots on the surface of them. So they always remind me of one of these lipid micelles. And I can just imagine each dot is a phospholipid head, and then the tails would all be pointing inside of my basketball. So this is how they typically behave if you were to just, throw some of these phospholipids into some water. They're going to hide the parts that don't like water and only have the parts that do like water exposed to the outside. So this works fine if the inside is full of air, right? It doesn't have any water, but this isn't going to be really functional for us for a cell. So what we see happen with these phospholipids is they're actually going to form what we refer to as a bilayer. So I'm going to go ahead and underline bi here. So bi means two, right? And so what we can see here is we actually have two layers, right, of phospholipids. And one side is going to interact, let's say, with the outside of the cell. I'm just going to put an O. And the other side will then be exposed to the inside of the cell. And all of the tails then are kind of pointing towards each other, creating this very hydrophobic region in the middle. So we've accomplished our goal that both the inside and outside of the cell can be exposed to water. We have those hydrophilic heads pointing there. And we have then this barrier with these hydrophobic tails that are going to prevent things from just passing through this barrier. Okay, does that make sense to everybody? So this is the basic construction of what every cell looks like. So if you were to look at a cell from a plant or an amoeba or a human and any of the gazillion types of cells that make up any of these organisms, all cell membranes are going to have this basic construction, right? You can think of this as the framing like you would of a house, right? We all have the same components. So over here on the right, this is one of my favorite pictures from our textbook. I think this shows really nicely what can pass through this phospholipid membrane and what really can't. All right. So notice that this is a log scale. So we're going up by a factor of 10 each time. The top means the stuff can get through this phospholipid with absolutely no problem at all. All right. So things like dissolved gases, which is what oxygen, carbon dioxide, and nitrogen are, they're going to be able to go back and forth across this membrane without any need for any assistance. They can just really get through here. And the direction will be based on how much is located on one side versus the other. All right, so we'll talk about diffusion here in a little bit. But the main point here is that dissolved gases are pretty much the only thing that can get through with such ease. All right, so they're able to get back and forth with no problem. Water's a tricky little thing, okay? So water, we know, is a polar molecule, right? Um, but it's also pretty small if you look at just an individual water molecule. So even though it's polar, very small amounts of water are able to get through the membrane without any specific protein carriers. Okay, so we will see some water be able to get through. You all are looking at some of this here in the next couple weeks in lab, right? You're looking at osmosis, which is the movement of water across a selective permeable membrane. And so this is how the water, it is able to travel through, but just in small quantities. We'll see how we can get large amounts of it through here in just a little bit.
I had a question
Yeah.
just about, I mean, it's relating to water here. It says that, O2, CO2, N2, small nonpolar molecules can go through pretty much without any restrictions, but small uncharged polar molecules. Does it say uncharged just because it's like not explicitly charged like an ion, but the molecule itself is still polar?
Oh, for
By
here?
uncharged,
Yeah, so let me,
what are
yeah,
we talking about?
small uncharged polar molecules. Yeah, so uncharged, so our textbook, thanks for bringing that up, Sebastian. Our textbook uses this term uncharged when it's talking about polar molecules. So charged is really reserved for ions.
Yeah,
So
actual
yeah,
particles that make
so
it.
these
Okay.
small partial charged
Yeah,
molecules can get through a little bit.
yeah.
Things like glucose and sucrose, even though it kind of shows it coming through, this is so small that we would basically say it's not getting through at all. You'll have just the slightest trickling, if that makes sense. This is a very, very low quantity, almost immeasurable. So you can see most of it gets bounced back. It doesn't go through. And then when it comes to ions, there's absolutely none of them that can get through without the aid of what we're going to see of a special protein carrier. So ions will never just drift through the membrane, where you may occasionally have a molecule of glucose or sucrose get through. But again, you can see most of it gets bounced back. And even with the water and glycerol here, a lot of it gets bounced back as well. So this is a good chart if you want to copy it and put it in your notes somewhere, just to help you remember what can get through and what can't. This is definitely something, as we're working through the semester, that's going to be very helpful for us to be able to recall pretty quickly. So we want to kind of commit this information to memory as best as we can. All right. So when we're looking at this phospholipid membrane, one of the models that has been put out there for quite a while for the lipid micelles. That's correct, Wesley. The chart is really just talking about the phospholipid membrane. Great question. Let me leave that chat open. Okay. So this fluid mosaic model is kind of how I want us to think about what these phospholipid membranes look like and kind of how they behave. So the first term here, fluid, is really important for us. It means that this membrane isn't rigid, right? So if we look down here, I know we haven't discussed all these components, but like we have some proteins, which are like these green and purple blobs. You have some cholesterol, which are these yellow things. We have some carbohydrates kind of sticking here up on the top. So there's lots of stuff that we'll see is embedded within these phospholipids. And the thing is this stuff can kind of move around laterally within the membrane. So it's not like a stiff structure, okay? The term mosaic means that we've got different parts, right? So there are lipids and proteins, so very distinct. You can identify them, right? Just as if you were building a mosaic out of different colored tiles, you can clearly separate proteins, sugars, and phospholipids from each other. The phospholipids themselves, we're going to see, are going to use our favorite van der Waals interactions to help kind of keep them in place and give it some shape. But these interactions, again, are going to allow the phospholipids to kind of move around laterally in the membrane. All right. Sometimes I think this is really hard for us to kind of conceptualize or visualize in our minds, because most of the things that we see, right, in real life are pretty, solid and don't have this ability to move. One interesting fact is to realize that if our phospholipid membranes were only made out of phospholipids and we didn't have these other components like proteins and sugars and cholesterol, this would be like olive oil. All right, so it would be very liquidy, it would just be sliding everywhere. So these other macromolecules that we're adding to this membrane are going to help to give it some structure, as well as different functions. So let's take a minute here just to look at the lipid composition. So again, we're looking at the phospholipid tails of our phospholipid membrane. So I mentioned that these chains can be of various lengths. So you can have long versus short chains. The longer the chains are, again, the more van der Waals interactions we're going to be able to have. So it's going to be kind of like butter, where it can pack really close together and form lots of interactions and make a pretty stiff um, membrane. Shorter chains are just going to have less opportunity, right, to form some of those van der Waals interactions. Um, the saturated versus the unsaturated also plays a big role in how fluid the membrane is going to be. So again, the more unsaturated, so we can see here they've kind of highlighted with these little boxes some of the double bonds or kinks that are located in these phospholipids. And so you can see it's really hard for them to get really close together in some areas. And so this is also going to provide a little bit more fluidity within the membrane itself. Does anybody know why am I even talking about fluidity of the membrane? Why, how this might impact the function of our phospholipids? If they're packed really tight versus not as tight? Does anybody want to just give out any thoughts about what that might?
Well, if they're packed tighter, won't that like restrict molecular movement through them?
Yeah, absolutely, Max. So yeah, the tighter they are, right, it's gonna be really hard even for gases and stuff, right, to get through if it's like packed super tight. So it is important that we have some of these gaps within here so that stuff can get through. It's also going to be important with regards to temperature. And so we talked about how cold temperatures, slow the movement of electrons down. You guys remember that? And so at the same principle applies here. As the temperatures drop, we would see the fluidity decrease and again, make it really stiff and hard for these membranes to be flexible, whereas warmer temperatures will increase it. All right, so it's going to follow the same behavior that we've talked about with just things like water and talking in general about electron movement. All right, so we want to have a nice balance of chain length, of saturation versus unsaturated. So again, that we can have membranes that are going to be usable in different environments. Right, so let's start putting some fun stuff into our phospholipids. All right, so we're going to start adding proteins. All right, so proteins are another class of macromolecule, and we'll talk about those, I think, at the end of next week, how we make them and things like that. But one of the important things to remember about proteins is that depending on the materials we use to build them, that they can also be hydrophobic and hydrophilic within the same protein itself. So we have these proteins here in the center that are known as integral proteins, or sometimes you may see them referred to as a transmembrane protein, which trans means that it goes through. So these transmembrane or integral proteins, we're going to see have both hydrophilic and hydrophobic regions. And therefore, they're able to interact with inside and outside the cell, as well as within the phospholipid tail component of the membrane. Right, so we're going to see that these integral membrane proteins are going to be critical for the passage of some of those molecules that can't just come through, right? So glucose, water, and our ions are all going to take advantage of proteins that are able to span the entire width of our membranes. We will see some anchored membrane proteins. These are going to be covalently bonded to the fatty acids, as well as some peripheral membrane proteins, which are kind of like these blobs you kind of see here on top. There's one here and there's one here as well. And these may act to identify what type of cell it is, or they may be working in conjunction with the integral membrane protein as like a key that can open and close the opening there. They can also attach the cell to other cells or cause reactions to occur within the cell. So lots and lots of functions with proteins. But the key to remember is that, the part that they can be hydrophilic and hydrophobic, depending on which portion of the cell membrane they need to interact with. So they're very, very specific types of molecules. All right, so just a quick review here. So lipids are a diverse group of molecules, primarily made up of carbon and hydrogen. They don't like water. All right, so as long as we can at least keep that big picture, and that will help us. They are involved in energy storage, thermoregulation. We talked about pigments, so light absorption, and we're going to see that some of that intracellular communication. We talked a little bit about steroids, right? And steroids are a hormone, so they're a form of communication within the different organisms. And we're going to see that they are extremely important, right, to many of these biological structures, including the phospholipid membrane. All right, I'm going to leave you guys. These are just some questions you should be able to answer, but just for the sake of time, because I do want to try to get through as much as we can today of this presentation, do take some time to go back over this presentation and, ask yourself, what is the difference between a fat and oil in terms of their structure and then how they're able to function? And then, what is special about the phospholipids that allow them to form bilayers, but triglycerides cannot? So a hint here is look at their structure. How are they built? And how does that allow for the formation of bilayers in one and not the other? And you're welcome to post your answers to these in like the Ask It basket, and then you guys can communicate with each other. I can chip in. Just a good place to kind of have those discussions. All right, you guys ready to move on to talking about the transport part of this? Anybody have any questions before we move on? Trying to see if I see any. Okay, Max is saying he's good. Just kind of flipping through, see if I have any questions. All right, looks like everybody's either asleep or good to go. All right, so transport now. So now that we've built this lovely structure that nothing can get through basically except dissolved gas, now I want to be able to allow things to go back and forth. So we're going to define what a selective permeable membrane is. We're going to distinguish the difference between a simple and facilitated diffusion. All right, so we're first going to have to define diffusion and then what we mean by simple and facilitated. And we will also look at osmosis, which again is going to be a topic that you guys are working on for about 2 weeks in lab. So we'll talk about it some here and you'll talk about it more there. And I'm also going to look at, I forgot to put a learning objective on this slide, at active transport. So we'll compare passive versus active. One is going to require energy and the other one isn't, which is also a very important distinguishing characteristic and also really essential for how cells are able to operate. All right, so selectively permeable. So what this means, or do you guys want to, how about someone give me what they think selectively permeable might mean? So I tell you it acts like a two-way filter. When I say that, what does that mean to you? Shannon, do you want to give a go at what that might mean? Okay, Riley put in the chat that only allows certain things through. Yep. Okay, so we have some good answers coming through, a couple in the chat there. So yeah, selectively means, right, that only certain things are going to get through, and permeable means it can get through. All right, and so we talked about that already. I showed you that graph, that figure. Passive transport. Passive means that it doesn't really require energy. Okay, so no energy required. We haven't talked a lot about physics yet in this course. We will get to some of that in our next theme. But what this means is that molecules are going to basically flow from an area where there's a lot to an area where there's less. All right, so that we can establish an equilibrium. So if we were to look at physics and look at how the whole, system here on Earth works, it's all to get basically to a nice equilibrium. And so passive transport basically means that stuff moves from an area of high concentration to an area of low concentration, and that movement doesn't require any energy. The converse of that then is active transport. And so the term active should make us think of energy. So requires energy. All right. So I always think of the junk closet, like in your house, right? That is just so overflowing that if you open the door, like stuff just falls out. But if I want to put more in there, right, it takes a lot of energy to shove it in, right? So the passive transport is all the junk falling out. The active part is me expending energy to push it back in. All right, so if you kind of have that type of visualization in your head as we talk about passive and active, I think it helps. All right, so down here I just have an image kind of showing us some of these different transport mechanisms. All of the ones I'm showing down here are passive transport mechanisms. So here we just have some simple diffusion where stuff is able to just pass through the membrane. So this would be things like oxygen and carbon dioxide. Over here, we've got these special proteins, and we're going to talk about each of these that allow specific molecules to come through. And then over here, a very special one that we're going to talk about a lot is the aquaporin, which is a special pore just for water. So you can see here, some water's getting through the membrane unaided, and over on this side, we see that we can get more water to come through using this protein pore. All right, so let's define diffusion before we get too far into things. So diffusion is basically the random movement of substances or particles, again, from an area of high concentration to an area of low concentration. So again, from high to low. So if we look at this really simple diagram over here, you can kind of think of this as, a cup of water and you're making some Kool-Aid or something, right? And so you threw in some red sugar particles. This represents a high concentration. Now, just because it's the way that things work according to the laws of physics, this is not going to want to stay as a clump. It wants to be uniformly dispersed within this fluid. And so over time, we would see that these molecules are going to disperse evenly throughout this container of water. All right, does that make sense to everybody? So this doesn't take any energy, it just takes time, right, for it to go from this state to this state. And so that is considered diffusion. All right. Another thing you can look at, is like if you spray air freshener in the air, right? It's diffusing from that point of release throughout a room, right? So it diffuses across the area, all right, with the goal of creating an equilibrium. Like rolling down a hill, high to low. Sort of, Dwayne. That's moving from potential to kinetic energy. So yeah, gravity's in play there. So yeah, we could kind of think of that as well, the energy to roll. The other thing I want to bring up briefly here, and again, we'll spend more time talking about physics and stuff in our next theme, but this second law of thermodynamics, which is one of my favorites, has to do with how the system that we live in here wants to go to a disordered state known as entropy. All right, so entropy, you can equate that to kind of chaos or disorder. And so again, if we compare the beaker on the left to the beaker on the right, we see that this is more organized, right? I got one single clump of molecules. We're here now, it's kind of all dispersed. So this is more disordered. So we went from an area of low disorder or low entropy to an area to a state of high disorder or high entropy. So again, this is the natural flow, natural process of things. In order to get this back to an organized system, I would need to apply energy. All right, so again, we'll talk more about that second law of thermodynamics, but I just want to kind of introduce it now to help us think about, the way things like to move. They like to create this state of equilibrium, which usually when we're talking about molecules and fluids, we see that going into a very disordered state. So there are several factors that are going to influence the rate or how fast, right, this diffusion can occur, right? So temperature, the concentration gradient, and also the area, the amount of space that a particular substance needs to cross. So let's kind of unpack these a little bit. So temperature. Do you think increasing temperature or decreasing temperature is going to allow for diffusion to happen faster? Would I want to increase or decrease the temperature? All right, I'm seeing lots of answers come in. Very good. So we're seeing increase. Does anyone want to explain why it's increasing?
Would that be because the particles move faster as they are heated?
Yes, Kristen, very good. Yep, the particles are moving faster as the temperature goes up. So as they're moving faster, right, they're able to spread out and reach that equilibrium faster. Does anybody know what I mean by a concentration gradient?
Isn't like that like how concentrated something is in a solution?
Yes. Versus, I guess, the other side. So if we were, let's just draw a little picture here. So let's say I had this goofy beaker that I got a discount because it's really wonky looking. All right. And let's say that this beaker had a membrane kind of separating one side from the other, right? And then let's say, oops, where'd the colors go? All right, let's say on this side, I had, what is that, 5 particles. And let's say over here, I had 6. All right. The gradient is kind of the difference, right, in the concentration from one side compared to the other. So would you think this is a high concentration gradient or a low concentration gradient? Or a difference, I should use maybe that word. Do you think there's a big difference in the concentration gradient here? I see a couple of people shaking their heads. No, right? This is pretty similar. So we don't have a huge driving force, right? But if I put a lot of particles on this side, right? So I have a much higher concentration on this side of my beaker than over here. This would be considered a bigger difference in gradient. And so we would see that there's a larger driving force that's going to push these over here to establish equilibrium. All right, and so that driving force can help increase the rate of diffusion. It will happen quicker. Does that make sense? We're going to use this term gradient a fair bit. So I want to make sure everyone kind of has at least a bit of an understanding that we're looking at the difference in concentration from one side to the other. All right, and typically we're going to be looking at a cell, and we're looking inside the cell versus outside the cell. All right, so that's what we're going to look at. So if this was a cell, you'd say, okay, this stuff wants to come in here to make an equilibrium. All right, and then the area which it has to cross. So if you're thinking about, how far, if this membrane was really thick, right, that I had in here, I know it's the wrong color, but that's okay. We'll just make it a really thick membrane. It would take longer, right, for those things to get through. Or if I was even inside of a cell, if the cell was huge, right, it's going to take a long time for something to get from one side of it to the other. So distance that it has to travel is really important. All right. One thing I do want to point out, right? Many of the solutions that we look at in a biological setting often contain more than one solute. All right. A solute is the substance that you're dissolving in the solvent, right? So I didn't really define these terms. So a solvent is usually going to be water, right, for biological organisms. Solutes are going to be anything that we're adding to that water. So it could be proteins, sugars, ions, dissolved gases, all right? All of those things are considered solutes. When we're looking at diffusion, we have to look at each solute on its own, right? So if I had sugar and sodium ions and proteins in this messy mix over here on the left, I would have to look at the diffusion of each of those solutes on their own, because how they're going to travel depends upon their concentration as well as some of their other factors, right? So just keep that in mind, that we're going to look at each thing individually, even if you have a whole bunch of stuff, because that's real life, right? If you look inside your cell, it doesn't just contain, one protein or one type of sugar molecule, right? It's got lots of stuff in it. And so this is a really important thing to kind of keep in mind. All right, so here, this was just a nice picture that I found that I thought showed kind of this diffusion. And again, we could think of these as dissolved gas molecules since there's no proteins helping us. So if, say we had a bunch of oxygen, right? So let's just say that these are O2 molecules on this side of the cell membrane and none inside of the cell, we would see that the oxygen is going to want to move in this direction, right? From an area of high concentration to an area of low concentration. And so over time, we start to see those oxygen molecules migrate from outside the cell to inside the cell until we've established an equilibrium. All right, now we're going to see that these oxygen molecules can actually flow back and forth at an equal rate. So we would never notice a difference in concentration once it has established an equilibrium. All right, does that help? I like Kristen's picture because she's always got these big thumbs up. I'm like, all right, she's got it. So this is what's happening with diffusion, all right? This is happening without any energy. It's passive. It's simply just following the laws of physics, going from high concentration to low concentration until we establish equilibrium. All right. So here's a question. I'll give us just like a minute or two to kind of think about this. So diffusion typically is a very slow process, especially in living tissues, right? Except if they're only crossing a really short distance, right? So it would actually take several years for a single molecule of oxygen gas to diffuse from your lungs through the tissues in your body to reach your cells in your fingertip. All right, if we hadn't evolved this particular system in our body that allows for faster distribution, can you think of what that might be? So if they just had to diffuse from the tissue in my lungs and had to get to my fingertips, right? By going from one thing to the other, you're on the right track there, Ben. What is the blood traveling in? All right, so I'm seeing some veins, the cardiovascular system. Yeah, so absolutely. Um, so Ben mentioned blood. So yes, blood picks up the oxygen molecules, right? But blood itself has to travel through, Dwayne, you got it, the oxygenated, so the arteries, right? In our cardiovascular system in particular, right? And then those blood cells, once they drop off the oxygen, the vessels then are named what, veins, and it takes it back up to the lungs where we can replenish it with more oxygen, right? So we're using the cardiovascular system to transport the oxygen to help it kind of get from the lungs, right, to all of the tissues in our body in a reasonable amount of time, right? Because if it had to diffuse from one cell to the next cell to the next cell through all the tissues in your body, it would take too long, right? The cells would essentially die. Plants also have a vascular system that allows them to transport molecules in an efficient way. Good job. All right. So we've talked about just simple diffusion, right? Which was the image I just showed us with the oxygen molecules, you know, coming right through that phospholipid membrane. So we're going to define that as simple diffusion. They don't need a protein to help them. All right, these molecules can simply pass through the membrane based on their concentration gradient. So they'll move from an area of high concentration to an area of low concentration. And again, those particular molecules tend to be very small, non-polar, uncharged, hydrophobic, basically dissolved gases are what we see here for simple diffusion. What we're going to see a lot more of is what's termed as facilitated diffusion. So the term facilitate means, right, that you're helping, you're assisting with the process. So we are going to assist the process of diffusion. So here we're going to use a couple different types of proteins. So there's a channel protein, which I like to define as a doorway, right? So we have different size doors for different purposes, right? So you have what we call like a person door, right? That people can walk in and out of. You have garage doors, right? That cars can drive in and out of. So channel proteins are like doors. It's going to be based strictly on the particle size. So as long as it fits through that channel, the particle or that molecule can move through. Carrier proteins are going to be a lot more specific. They actually have to bind to the specific molecule in order for the molecule to pass through. So if we look at the image here on the left, you see a channel, which is kind of just like a chute, right? And as long as the molecules can fit through this opening, they're going to be able to use this channel to go from one side to the other. And notice it's going from an area of high concentration to an area of low concentration. Where a carrier protein is based on shape. So we can kind of see that it kind of conformed to this round molecule here. So in reality, the shapes of molecules are a lot more complex than little round circles. So these are going to be very specific for whatever type of molecule that they're allowing to pass through. But again, it's still diffusion, which is passive. So we're going from an area of high concentration to an area of low concentration. All right, both of these types of protein channels and carriers are going to transport large polar charged hydrophilic type of molecules. All right, so they can, depending on the application, we can assign different things, different molecules to these different types of proteins. All right, so these would be considered those transmembrane proteins or those integral proteins because again, they're spanning through the entire thickness of those phospholipids. So like a door that needs a key. Ah, Riley, you're getting ahead of me. Yes. Sometimes we'll see that these channel proteins or even the carrier proteins are going to need a key to open them. And it won't be just based on concentration. So sometimes the doors are just going to be open. All right, we live in a safe neighborhood that we don't have to lock our doors. Other times we're going to see that we do have to control it a little bit more, and we'll have to find a way to open and close it. Great analogy. All right, let's see. So it actually takes us right to our next set of slides. All right, so here are what we're going to call gated channels, which is what Riley was talking about with something needing a key. And we're going to look at two different types. We're going to look at voltage gated channels, and we're going to look at ligand gated channels. All right, so this first one here is voltage. So this is kind of a weird key. This is going to be based on charge. Okay, so voltage is associated with charge. And what we're looking at with charge is the difference in charge between the inside and outside of the cell. All right, so I guess this is the inside usually. And this is the outside. All right. And so what happens is that there is a electrical potential, right? A difference in charge between the outside and the inside. And that difference is what can open up some of these ion channels. So notice all of these examples I've given you here are ions. We've got sodium, calcium, potassium, chloride. So we've got positives, negatives, +1 charges, 2+ charges. And so based on what the charge is out here will depend on whether or not the specific ion channel will open or close. All right, this is a pretty complex system. One of the applications where we study this a lot is in the nervous system when we're looking at like creating action potentials and things like that. Will open up a bunch of these different channels. So those of you who have taken physiology, I don't know if the my exercise science students, if you all have taken your physiology courses yet, but this is one application where you're going to see a lot of that. In this course, we don't go in quite into that depth, but we will talk a little bit more about this charge difference. All right. So again, we have to always think about ions as carrying that charge. The second type has a funny name, all right, but again, it's gated, and they're called ligands. And really, a ligand is a very general term used to describe any type of molecule that binds to a protein in this case, okay? And basically, this is going to act like a key. And what it does is this particular molecule will bind to the actual protein carrier or channel and open or close it. All right. In this case, we're looking at a cell membrane responding to a signal from the nervous system. So we have a neurotransmitter, which is a chemical that has a specific binding site on this particular protein channel. And when it binds, you can see that it goes from closed to open and allows this specific ion now to pass into the cell. All right, so this one really does work like a key. So again, these are ligand gated channels. And again, it's allowing some control over what can come in and out of the cell. So we have to have something telling this to open. It wasn't enough just to say, oh, I've got a ton of ions out here and none in here. It actually has to be told to open. So it's a level of control. And I think I am out of time. So I'm going to go ahead because I do want to respect all of you and your schedules. What I think I'm going to do, if it's all right with you, is I'm going to go ahead, and if you want to stay on, and you can, I'm going to finish lecturing through these last few slides. And then you can go back and just listen to the end of the presentation if you can't stay. But if you can stay, we'll wrap it up so that we can stay on track for the rest of the week. Does that sound like an okay plan for you? Does that, I see, okay, I see some thumbs up. Okay. So I'm going to go ahead then and just continue, and you're welcome to stay. And if you need to go, again, you're welcome to leave. So I will post this as soon as it's done processing. Okay. So let's go back to our presentation. What we're going to talk about now is osmosis, which is a little bit different than just plain diffusion. Okay, so here we're looking at the movement of water instead of the movement of the solutes. Okay, so what we're going to see is that in certain situations in living organisms, we cannot establish that equilibrium that we want to by moving the solute. So by moving the salt or the sugar or the protein, we have to move the water because the membrane at that point doesn't have the right channels or whatever to allow the movements of those solutes. So what we're going to use is this process known as osmosis. All right. So again, osmosis is the movement of water. And that's really, really important to kind of ingrain. We're going to pull in a special protein carrier known as an aquaporin. All right, so these are specialized channels that are only going to move water molecules. So I found this kind of cute image here this morning that I really liked. So these are our little water molecules. Here's our phospholipid membrane. And then this green structure here that you see is an aquaporin. All right. And a way to remember this is aqua means water, and porin is referring to a pore. So this is literally a water pore that can appear in our cell membranes. And the only thing that can pass through it is water itself. Okay, Tyler had a quick question. So since the smaller cell, they help to activate the channels between cells. I will get to your question in just a second, Tyler. I think I'll clear that up, and we'll talk about active transport here in a second. All right, so with osmosis, here's where things tend to get confusing. What we're looking at again is the movement of water from an area of low solute concentration to an area of high solute concentration. All right, so what am I talking about? I'm talking about dilution, really. All right, so let's just go ahead and add that term here because I think that helps students and even faculty sometimes when we're talking about osmosis to remember what's happening. So if we look at this picture over here on the right, let's pretend I have a bunch of sugar molecules over here. Okay, so this blue stuff is sugar. And let's say I only have 3, okay, on this side of my cell membrane. Normally, what we would have seen, right, is the sugar would move from an area of high sugar concentration to this area of low sugar concentration, right? That would be diffusion. But I don't have a protein carrier or channel to allow the sugar to move. So in order to establish equilibrium, the water has to move. So in order to get the concentration of the sugar to be the same on both sides, I have to dilute this sugar down here by adding more water. So the water's flowing in this direction, right? So the water's coming down here and diluting this solute. All right, so if we go back to our statement of osmosis, we're looking at the movement of water from an area of low solute concentration to an area of high solute concentration. Does that make sense? So you're diluting the sugar, right? We want it to be the same concentration as up here. So to do that, I have to add more water. All right. So when you're thinking about your experiments with your eggs, especially in part 2, because that's the part that's dealing with osmosis, and you're working with the sugar component, right? Think about which side that sugar solution is on, and think about which way the water's going to move, right? Because your eggs are basically made up of cells with these phospholipid membranes. Think about which way the water's going to move to help establish this equilibrium. Okay, so wanted to kind of give you some context there. These other terms, osmotic pressure and osmolarity, can be a little bit confusing, but let's see if we can try to define them in a way that makes sense. All right, so osmotic pressure is the pressure that needs to be applied to a solution, all right, so something dissolved in water, to prevent the flow of water across a membrane by osmosis. All right, so this one, it's a little bit hard to, I think, visualize when we're looking at a very simple picture like this. Where this comes into play a lot is when we're looking at our capillaries, because we have pressure that's being exerted from the outside onto the capillary, and pressure that's being exerted on the inside of the capillary wall from like all the blood and stuff that's passing through. All right. And so you're looking at the pressure of that's being applied that's going to prevent the water from going from one side to the other. I am going to just tell you right now, I am not going to test you on osmotic pressure or have you determine what that pressure is or which side is higher or lower in this class. We don't go into that level of detail. If we had more time to get into human physiology or animal physiology, we could expand on this. But for now, just understand that it's pressure that kind of keeps the water on one side of the cell membrane. The osmolarity is the total number of solute particles per liter of water. So if we wanted to know the osmolarity for this system, we would have to actually measure it outside the cell and inside the cell to get that figure. Again, I'm not going to have you calculate that in this course. I just wanted to define that term for you so that you had a rough idea of what it was talking about. It's just a way that we can measure concentration in this type of situation. All right. All right. So let's take a look here at where we really do apply osmosis. All right, so our textbook is using, and most textbooks use this, are red blood cells, right? So this is an easy thing for us to visualize. But I want you to understand that this process happens in all of your cells, all right? And that's what the purpose of the case study and stuff is that you're working on in lab this week. You're going to see how this actually applies to all of our cells. But here in lecture, we'll focus first on looking at these red blood cells. So there's a term here called tonicity. All right, and tonicity just basically means that I'm going to compare two solutions that are separated by a selective permeable membrane where we can see osmosis take place, right? So kind of a very specific definition, right? So, oops, that's not what I wanted to have happen. All right, so up at the top here, what we have are three terms: hypertonic, isotonic, and hypotonic. So can anybody remember from week 1 what the prefix hyper refers to? I know I'm having us go back already 3
Over,
weeks.
I think.
What was that, Dimitri?
Over, no?
Yeah, over, excessive, exactly.
Yeah.
So we can, yep, both of those terms work. So over, excessive. All right, I always think of someone who is hyper, has excessive energy, right? So if you're hyper, you've got excessive energy. What about hypo? This one we don't use a lot, I think, in normal everyday language, where like we would the word hyper. What about hypo?
That'd be
Tyler?
like,
Not enough.
Do you want to go ahead, Naira?
Oh, I just said, is it under?
It's under. Yeah, it's under, low. Yep. Okay, so, and then, whoops, just scribble that out. Forgot the L. All right. And then what about ISO? What do you
Same.
think? The same. Okay, so these three prefixes, super, super important to remember, especially when we're talking about osmosis. Okay, so if you don't know these already, just like hyper means more, hypo means less, ISO's the same, put some flashcards together maybe and just quiz yourself on it. Okay, what we're talking about is the solution that the cells are in. So whenever we're talking about osmosis and I say hypertonic solution, I'm talking about that the solution that I've put the cells in is hypertonic. Meaning, if we look at this bottom picture here, that there is more solute. I'm just going to put a plus sign for more, okay? More solute outside the cell. Okay, outside. Okay. So if I am in a hypertonic solution, that means that I have more solutes outside than I do inside. If I'm talking about osmosis, only water can move, right? So you see here, these arrows are showing us that the water is going to leave the cell and kind of dilute this environment outside of it. Does everyone see that? So if a cell is in a hypertonic solution, the cell will lose water to that outside environment. All right. The same thing happens when you swim in the ocean, right? So hopefully if you've ever been to the ocean, your parents or guardians told you to hydrate frequently, because what's happening is there is more salt, right, in the ocean than in our body, in our cells. And so our cells are trying to form an equilibrium with the ocean water, right? Who's going to win that battle, right? The ocean. You are going to dehydrate because the ocean water is going to cause your cells to lose water through osmosis. All right. And that's why you kind of get your skin gets really dry and things like that after swimming in salt water. The ocean is a hypertonic solution. All right. Let's jump over here to the far right where we have a hypotonic solution. So here we have less solute outside of the cell. All right. And so in this situation, we're going to see that the water is going to want to move into the cell to dilute this. So they didn't really, let's put some more solutes in there. They didn't really put enough. All right, so this is going to move in. All right, and notice what it did to these red blood cells, right? They're starting to get bigger, and we see that eventually that it can cause cells to explode. All right, so some processes will happen inside the cell due to the influx of water, and it will lead to the cell actually exploding. So this is a hypotonic solution. So those of you who going into the medical field or things of that nature, when you think about You know, why wouldn't you give a burn patient or someone who's dehydrated just plain H2O in an IV? It's because it would cause their red blood cells to explode, right? If we want to keep everyone's blood happy and they need fluid replenishment, you need to make sure that the fluids you add is isotonic to the concentrations of what's actually in our blood. So there is some sodium in there, right? So you would need to match that so that the cells stay happy. All right, so in isotonic, you have an equal movement of water in and out of the cell such that you're not going to see any net change. In a hypertonic solution, the water's going to leave the cell, cause it to shrivel. In a hypotonic solution, water will enter the cell, causing it to swell and possibly burst. All right, I highly recommend you do some practice problems with osmosis. There's usually some questions on quizzes and tests. And draw beakers. I always draw a beaker and model whatever the situation is. So you can put a little cell in, and if it tells you, you know, you've got 1 molar of a solution out here and 5 molar sugar solution in there, you know, which way is the water going to move through osmosis to establish an equilibrium? What would you guys think? Is it going to move towards the cell or away from? So towards or away? Which way would the water move? Okay, so I'm seeing a lot of away. So let's define, so again, this is water in this beaker, and it's at a 1 molar, let's say, sugar solution, and inside my cell is a 5 molar sugar solution. Is this representing a hypertonic environment or a hypotonic environment? Let's establish that. It's hypo, right, Carrie, good. So this is a hypotonic, right? Because I have less or a lower amount of solute outside my cell than I do in. So which way now, since I know it's a hypotonic solution, which way is that water gonna flow? Ben, I think you had, it's gonna flow inside, right? So the water's going to go this way, right? Because the sugar doesn't have a transporter. So only the water's able to move in my situation here. So water has to move into the cell to dilute this until they're both at the same concentration. All right, so you can create a bunch of different problems like this, changing the numbers and practicing identifying, is it hypotonic, isotonic, hypertonic, and then figuring out which way the water's going to move. Okay, I think that would be really, really helpful. All right, I think I have kept you guys long enough. So I still have active transport, but I don't want to drag this on too long. So we will finish up active transport. I'll look at how long our IRAT TRAT is. I think we might have time to talk about active transport before that on Wednesday, and then you guys can take the quiz. So I'm going to stop here. So that I don't oversaturate your brains with knowledge transport, right? I think our transporters are overwhelmed. So I will stop here. If you have any questions,